programa de pós-graduação de clínica médica, e a gente tem o imenso prazer de receber hoje aqui um convidado internacional, o professor Samelson-Jones, que é professor assistente de pediatria no Children's Hospital of Philadelphia, na Pensilvânia, nos Estados Unidos. O Ben já trabalha há um tempo, sobretudo com hematologia benigna, focado muito em hematologia, e o tema que ele vai discutir é Enhancing Coagulation Factor 8 and 9 for Hemophilia Therapy. Então, Ben, it's a great pleasure for us to have you here with us. We're really looking forward to your presentation. Please, the floor is yours.
Well, thank you so much for inviting me. I'm really excited to talk about our work and work by others improving factor 8 and factor 9 for hemophilia therapies. And my talk will mostly focus on gene transfer approaches, but these concepts have relevance for any hemophilia treatment. I have a number of relevant disclosures, including clinical research support to my institution and laboratory support from several companies involved in gene therapy for hemophilia. As many of you are probably aware, hemophilia is the most common severe bleeding disorder associated with spontaneous hemorrhages and excessive bleeding from trivial injuries. Before modern medical intervention, severe hemophilia was a lethal pediatric disease, with most patients dying in childhood, often from minor injuries that never achieved hemostasis, such as this picture from the early 20th century of a young man with a tongue laceration. Hemophilia is also uniquely characterized by a preponderance of muscle and joint bleeds, as shown here, that without therapy leads to debilitating hemothropathy, as shown on this picture on the right. At a molecular level, hemophilia is due to a biochemical defect in the coagulation cascade, which results in insufficient thrombin generation and thus poor clot formation, like you saw in the young man in the previous slide. Importantly, activated coagulation factor 8 and factor 9, circled here, work together to enzymatically activate coagulation factor 10, which is the rate-limiting step of sustained thrombin generation. Hemophilia is due to a deficiency in this 8A, 9A complex, and the bleeding is due to insufficient activation of factor 10. Hemophilia A is due to a loss of function in the factor 8 gene, while hemophilia B is due to loss of function in the factor 9 gene. They are both X-linked disorders, such that the majority of patients are male. Hemophilia A, factor 8 deficiency, occurs in about 1 in 5,000 males, while hemophilia B occurs in about 1 in 30,000 males worldwide. About 30% of cases are de novo, and hemophilia occurs across all ethnic groups. The bleeding phenotype of hemophilia is probably identical between hemophilia A and B because they both result in the same loss of factor 10 activation. The bleeding phenotype of hemophilia is highly dependent on the amount of residual factor, where patients with severe disease have less than 1% normal factor levels and have frequent spontaneous bleeds, while those with moderate disease have between 1 and 5% normal factor levels and bleed occasionally with trivial injuries, while those with mild disease between 5 and 40% normal typically rarely bleed, usually with major trauma or surgery. And this close relationship between residual factor and bleeding phenotype makes hemophilia highly amenable to therapies, including gene therapy, because small increments in the factor level can have a major clinical impact with changing the phenotypic severity. And the idea is for therapeutic interventions to move patients with severe disease into the moderate or mild phenotype range, or potentially into curative levels above 40% normal factor level. Currently, we mostly mitigate the complications of severe hemophilia by converting the disease from a severe to moderate or mild phenotype by the regular scheduled intravenous infusions of factor product replacements. though non-factor therapies, as I will mention, are becoming more popular. Here is shown the sawtooth-shaped graph of a typical factor prophylaxis, time to prevent the patient from falling below 1% normal activity. The basic idea of enhancing factor VIII or factor IX is that wild-type proteins may not be optimized for therapeutics. And the illustration of this rationale with the most examples in hemophilia care is the development of extended half-life factors that have been engineered with half-life extending moieties or domains for both factor VIII or IX, though to date this has been more successful with factor IX with half-lives several-fold longer, as you can see in this last column. This may be changing with a new engineered factor VIII BIV001, which was recently published in Blood from this year. So extended half-life factors have the ability to decrease the number of infusions needed per week for prophylaxis and potentially achieve higher trough levels, which is a practice that's being adopted for extended half-life products, factor IX products, as you can see here. At our Center for Hemophilia A patients, extended half-life products, mostly a lactate, which is the Fc fusion factor VIII molecule, did decrease the average number of doses per week from 3 to 2, but you can see that in both cohorts, there's still a wide range. And so the first and main example I would like to discuss is the use of factor IX podula for hemophilia B gene therapy. Gene therapy has the potential to deliver this hemostatic protection with a single administration of a gene therapy drug, offering the promise of many years of factor level, ideally in the curative range. And over the last decade, there have been several clinical trials demonstrating the potential of gene therapy for hemophilia B using adeno-associated viral vectors, AAV vectors, with wild-type factor IX. AAV has emerged as a highly attractive gene delivery platform, both for hemophilia as well as other genetic diseases, though AAV has its own pros and cons, as I will discuss. The first sustained therapeutically relevant factor IX levels after gene therapy for hemophilia B were achieved in a clinical trial sponsored by St. Jude's and University College London. Here they tested an AAV serotype 8 vector with a hepatocyte-specific promoter that expressed wild-type factor IX. AAV8, factor IX, wild-type. Shown in this graph are the sustained levels after gene therapy, and I've highlighted the thresholds to discuss. So here is 2%, which is the inclusion criteria of this study and most of the subsequent ones, the 5% normal level that demarcates moderate and mild hemophilia, and finally the curative level at about 40%. I've also included more recent clinical data evaluating a similar vector, but with an AAV serotype 5 vector. Importantly, there were no major safety concerns in either trial. As you would expect, based on the factor IX levels in the low mild range of 5 and 6% normal, the annualized bleeding rate, ABR, decreases in the gene therapy recipients, though most recipients still had about 1 or more bleeds per year. The difference in the pre-gene therapy bleeding rate between the trials is just related to the number of trial participants that were on factor prophylaxis prior to gene therapy. And I would emphasize that these factor IX levels of around 5% in the AAV8 trial have been sustained now for more than 8 years, as reported in meeting abstracts. There are several limitations on the administered AAV vector dose that can be safely and efficaciously given, including T-cell mediated immune response to the AAV capsid, AAV-associated hepatotoxicity, the risk of vector genome integration and resultant genotoxicity, as well as the manufacturing cost. However, as is true as a general pharmacological principle, the most practical approach is the use of the lowest effective dose. And these ideas then raise the question about how to achieve higher factor IX levels and thus further decrease the bleeding rate without increasing the vector dose. And the most successful strategy to do this, to enhance gene therapy for hemophilia B, as I will discuss, is the use of the gain-of-function factor IX variant, factor IX Padua, initially identified in part in 2009 by my mentor, Valder Ruda, as the cause of this rare X-linked thrombophilia. The proband of this disorder was a 23-year-old young man with a lower extremity deep vein thrombosis from Padua, Italy, who presented with this deep vein thrombosis, but without any other identified thrombotic risk factors. He was, however, noted to have an 8-fold elevated factor IX activity with a normal factor IX antigen level, such that his activity to antigen ratio was 8-fold normal, or an 8-fold higher specific activity. His younger brother also showed a similar increased specific activity, and their mother had about half that increase in specific activity, suggesting that she was a heterozygous carrier of this X-linked disorder. His father and other brother parameters were within normal limits. And based on this pedigree here, a mutant form of factor IX was identified with a single amino acid substitution of the leucine substituted for the arginine at position 338, which is termed factor IX Padua. And we've carried out a series of experiments using recombinant factor IX wild type, WT, and Padua to define the mechanism of this 8-fold increased specific activity. Here we observe in a plasma assay comparable to an APTT measurement that factor IX Padua has 8-fold higher specific activity than wild type, consistent with the observations from the original family. In an isolated protein system that just includes activated factor IX, factor X with phospholipids, where we look at the amount of factor Xa generated, we observe that Padua in red and wild type are similar until we add activated factor VIII as cofactor. In the presence of 8A, we see that Padua generates more Xa than wild type. We also found that the activation and inactivation of factor IX Padua and factor IX wild type are similar. And this enhancement in the presence of 8A seemed to be due to an improved allosteric activation. To determine if this improvement, this 8A dependent improvement was also going on in plasma and not just the isolated protein system, we developed a new assay that allowed for factor IX activity in plasma without factor VIII, utilizing a brand new factor VIII mimetic, Amicizumab, that had just been approved as a non-factor therapy for hemophilia A. What Amicizumab does is a bispecific antibody that complexes with 9A and factor X with sufficient geometry to allow for enough 10A generation for hemostasis. And for our assay, we blocked 8A activity with a cocktail of factor VIII inhibitors, such that all the 10A that was generated in the experiment had to pass through the 9A 10 Amicizumab complex. And what we observed that in regular plasma, factor IX Padua has the higher specific activity than wild type, purple compared to wild type. However, when we added Amicizumab to replace factor VIII, we observed that the specific activity of Padua was actually a little lower than wild type, supporting the idea that the enhanced activity of Padua is due to this improved interaction with 8A. We saw the same thing using a thrombin generation assay. Here we graphed the amount of thrombin generated from hemophilia B plasma with different amounts of recombinant factor IX protein. Here you can see that Padua, again in blue, results in much more thrombin than wild type in purple at any concentration of factor IX in this range. However, when Amicizumab is added, factor IX Padua in red and wild type factor IX in black appear almost identical over several orders of magnitude factor IX concentration, again supporting that the enhanced activity of factor IX Padua is dependent on its interaction with 8A. And these biochemical results support the safety of factor IX as a therapeutic, factor IX Padua as a therapeutic, since at a molecular level, factor IX wild type and Padua are regulated similarly and display the same 8A dependence. We also published the first reported use of Factor IX Padua in the Hemophilia B gene therapy in 2017, which has subsequently become the standard transgene for Hemophilia B gene therapy. This is what we refer to as the Robin Hood approach of gene therapy, when we take from those who have too much Factor IX activity, the Padua family, and give to those who have too little Factor IX activity, men with Hemophilia B. In the 2 published clinical trials, AAV-based gene transfer of Factor IX Padua achieved Factor IX activity levels in the curative or near curative levels, as summarized in this figure, and compared to the previous trials using wild type. These are the trials with Padua, these are the trials with wild type. Notably, as in the trials using Factor IX wild type, there were no major safety concerns. And though this figure would suggest that the AAV5 vector results in higher Factor IX levels compared to the Spark 100, I would just note that different Factor IX activity assays were used in these 2 studies. And we have recently showed that the measured Factor IX activity with Factor IX Padua has notable variation between different 1-stage assays. And this, which stresses the challenges of comparing the results between studies. What's shown here are gene therapy, are samples from gene therapy recipients expressing Factor IX Padua measured with 4 different 1-stage or chromogenic assay. And we saw a similar pattern between these reagents using recombinant Factor IX Padua. As expected, based on the natural history of mild hemophilia, the vast majority of gene therapy recipients with Factor IX levels in the high mild range or curative levels have 0 bleeds. Notably, this single participant with an ABR of 4 had horrendous hemothropathy with frequent pain and subjective sensations of joint bleeding, and was treated as such because we did not assume hemostatic efficacy of Factor IX Padua. He has subsequently had a knee replacement, and his ABR has decreased. So just to summarize the first part, Factor IX Padua has an optimized 8A interaction compared to wild type Factor IX. Adopting Factor IX Padua increases Factor IX levels after gene therapy without safety concerns. And using Factor IX Padua in AAV gene therapy has resulted in a near phenotypic cure of Hemophilia B. However, AAV vector dose-related concerns still remain. And that raises the question, can we do better than Factor IX Padua? Can we achieve higher Factor IX activities with less vector dose? I would just take a slight sidetrack here to note that there have now been 3 examples of thrombosis patients identified with Factor IX variants at position 338, 2 from the original Padua family, and more recently, a case report from Shanghai, China. We and others, including investigators at your institution, have now also evaluated over 767 additional thrombosis subjects without identifying additional variants at this position. And overall, then, the crude estimate of the prevalence of this type of thrombophilia is consistent with the rate of single nucleotide changes, as well as these Factor IX variants being strong thrombophilias. And so based on the results of the biochemical studies suggesting that human Factor IX wild type formed a suboptimal interaction with Factor VIII, we found it very intriguing that the arginine at position 338 was evolutionarily conserved among mammalian orthologs. And to better understand this evolutionary conservation, we performed site saturation mutagenesis at this position, substituting every amino acid into human Factor IX. Graphed here is the relative specific activity of all the variants compared to Factor IX wild type, which is the arginine shown in blue. And on the right is the activity and antigen of the different variants, also compared to wild type. We note that the leucine, the Padua variant in red, has the highest specific activity, supporting its widespread use in gene therapy. But a number of other variants, including the glutamic acid, which I've highlighted in purple, also have increased activity. Importantly, most of the substitutions at this position increase specific activity, suggesting that wild-type factor IX has not evolved to have optimized clotting activity. And based on that insight, we have identified a number of other factor IX variants with increased specific activity, which we presented at the ISTH meeting last year. And recently, there have been a number of other approaches to enhance factor IX activity for AAV gene therapy. This recent Blood paper uses a rational design, including the R33E, which we also found to have increased specific activity, to achieve modestly higher levels than factor IX PADWA, highlighted in this data in blue, after AAV gene therapy in hemophilia B mice. And another recent approach reconstructs extinct factor IX orthologs in silico and then measures their activity. One variant, when combined with PADWA, shown here in red, increases the achieved factor IX activity after AAV gene therapy in hemophilia B mice by about threefold, as you can see here. And I refer to you to those recent publications for the details. However, there's no such thing as a free lunch, and every amino acid substitution carries with it a risk of the immunogenicity of the new variant. And this concept is perhaps best illustrated by the history of two novel factor VIIa variants that were being developed as bypassing therapy for patients with hemophilia with inhibitors. They had 3 or 6 amino acid substitutions. Both of these independent engineered 7A variants resulted in anti-drug antibodies against the variant in their phase 3 studies with some cross-reactivity to wild-type factor VII. And importantly, these were in study subjects with normal levels of wild-type factor VII. In other words, the 7A variants broke the already established tolerance to wild-type factor VII. This is a very different situation and likely even less immune provocative situation compared to hemophilia patients receiving 8 or 9 variants. And to address this question of immunogenicity of factor IX PADWA, our group has carried out a number of studies evaluating the canine ortholog of factor IX PADWA in 2 hemophilia B canine models. This experimental system allows us to study the immune response to variant factor IX in a species-specific manner, which likely best recapitulates the patient experience. The models we use both have factor IX activity and antigen less than 1% normal and a severe bleeding phenotype that includes spontaneous bleeding, just like the human disease. The first model is from the Chapel Hill colony and is due to a factor IX missense mutation that results in misfolded factor IX protein. These dogs generally do not develop an anti-factor IX immune response after an infusion of canine factor IX protein, though they have developed inhibitors after muscle-directed gene therapy, which is a provocative immune challenge. In contrast, the model previously housed at University of Alabama have an early stop codon and no factor IX transcription. University of Alabama dogs are generally not tolerant to factor IX protein and develop inhibitors after a single infusion of canine factor IX protein. As such, we consider these animals inhibitor-prone. Here's an example of an inhibitor-prone dog developing an inhibitor as measured by Bethesda units in blue after a single infusion of canine factor IX concentrate. And so our group evaluated canine factor IX PADWA gene transfer in progressively more immunogenic scenarios, both compared to the target of gene transfer, muscle over liver, as well as the canine model, missense over nonsense inhibitor prone. We first tested the muscle-directed gene therapy in the missense dogs without observing an anti-factor IX immune response. We then tested liver-directed gene therapy in the inhibitor-prone dogs, including in a dog with a pre-existing anti-factor IX inhibitor. And most recently, we found that in the most provocative hemophilia B canine model with the most immunogenic target tissue, which we published in 2018, we observed that the canine factor IX PADUA and wild type have similar immunogenicity. Indeed, after gene therapy, these inhibitor-prone dogs tolerate infusions of wild-type canine factor IX protein. And combined, these studies support the continued use of factor IX PADUA in gene therapy with low risk of immunogenicity. And so it might be that PADUA is a free lunch, 8-fold higher activity with no immunogenic cost, which is why it's being used across gene therapy clinical programs. And I wanted to finish up by discussing work in optimizing factor VIII for gene therapy. I would emphasize that though the bleeding phenotype of hemophilia A and B are similar, factor VIII and IX are very different proteins, with factor VIII being over 4-fold larger than factor IX, which has complicated gene therapy approaches for hemophilia A. The domain structure of factor VIII is shown here, with the B domain in gray comprising about 40% of the protein. This single-chain form is initially translated, but then factor VIII undergoes proteolytic processing at the positions indicated by the green triangles by the intracellular protease pace-furin to form the heterodimer form, which is secreted into plasma. While circulating, the B domain undergoes further nonspecific proteolysis, such that the circulating factor VIII is a heterogeneous population of this heterodimer forms with variable residual B domains. Importantly, all of these forms retain hemostatic activity since the B domain is removed during thrombin activation. And based on this observation, the B domain deleted factor VIII was engineered. It was first used as recombinant protein, now under the trade name Xintha, and also was the backbone for extended half-life products such as aloctate. Importantly, even though it was not developed for gene therapy, it has generally been adopted to allow factor VIII to squeeze into AAV vectors under their packaging capacity. In the design of B-domain deleted factor VIII, these 14 amino acids were left in place after the entire B domain was removed, in part to retain the amino acids from 1645 to 1648, highlighted here in red, which comprise the canonical pace-furin recognition motif. This 14 amino acid linker is termed SQ because it connects the N-terminal B domain and the C-terminal at this SQ junction right here. However, the role of pace-furin on factor VIII biology had not previously been explored in detail. I would just add that this pace-furin recognition motif, highlighted again in red, has been maintained in most subsequent B-domain deleted factor VIII iterations, many of which are commercial products, as you can see here. And SQ is being used in most gene therapy products for hemophilia A, with the exception of VIII, which is a modification of the design constructed to enhance protein secretion as discussed in this blood paper. Yet, despite this widespread use, the role of furin processing had not been explored. And to study this, the role of furin in factor VIII biology, we designed a modified B-domain deleted factor VIII, where the amino acids comprising this recognition motif from 1645 to 1648 were removed, which we refer to as delta PF. We observed that stably transfected mammalian cells secrete almost 3 times more delta PF than B-domain deleted, orange compared to white, but that much more of the B-domain deleted, white here, remains within the cell compared to delta PF. This suggested that pace-furin processing is actually deleterious for factor VIII secretion, which is the opposite of what had previously been assumed, but not tested. We also found that this enhancement was dependent on functional pace-furin. We also observed that the amount of circulating factor VIII after AAV liver-based gene therapy in hemophilia mice at 3 different dosing cohorts. On the left here is antigen, on the right is activity. We observed that in the middle and high dosing cohorts, delta PF in solid circles displays increased expression compared to B-domain deleted, such at the high dose, almost 3 times more factor VIII is circulating after delta PF gene therapy than B domain deleted. And this increased circulating amount of delta PF was consistent with the previous in vitro observations that I showed on the last slide, and is likely due to this ability of the delta PF variant to evade the deleterious effects of paste furin processing. And we also observed comparable factor VIII levels after AAV gene therapy with delta PF in hemophilia dogs at a much lower vector dose compared to dogs that had previously received B domain deleted factor VIII gene therapy. Importantly, challenges with recombinant B domain deleted factor VIII did not elicit an anti-factor VIII immune response in these dogs expressing delta PF. These results from the large animal model highlight the potential of this novel variant to promote higher factor VIII levels after gene therapy. And more recently, in work in Lindsay George's lab, we showed that limiting factor VIII inactivation by activated protein C, abbreviated here as APC, enhances the hemostatic potency of factor VIII in hemophilia A mice. Here we designed a factor VIII with the two glutamines at the sites of APC cleavage. So while wild type factor VIII is inactivated by APC, as you can see in this cartoon, this variant, which we refer to as QQ, is resistant to proteolytic inactivation by APC. When these proteins are administered to hemophilia A mice, much less, about 5-fold less QQ is required to restore hemostasis compared to wild type in blue in a tail clip injury in hemophilia A mice. And this group is now working on studying if this enhanced hemostatic potency can be utilized to improve gene therapy for hemophilia A. I wanted to add that using optimized proteins for gene therapy is not unique for hemophilia gene therapy, and that several of the approved gene therapy products for other diseases are using such variant transgenes. This utility sort of highlights the importance of studying the basic biochemistry of proteins, which has long been my interest, which may yield, even decades later, as this hemoglobin variant did, therapeutically important results. And so I hope you got the following 4 take-homes from this presentation. Wild-type factor VIII and factor IX are not optimized as therapeutics. Both factor VIII and factor IX can be enhanced to improve protein and gene therapeutics. All current gene therapy approaches for hemophilia A and B utilize optimized transgenes, but the immunogenicity remains an important concern. I would like to thank the people in my lab, my mentor, Valda Ruda, my colleague, Lindsay George, and my other collaborators, as well as funding, and I'd be happy to take any questions.
Thank you, Ben. Thank you very much. It's very nice presentation, and I know all the work that you and your group have been work. So I don't know if someone wants to ask a question for Ben. Eric?
Hi. Hello, Ben. Thank you for the presentation. Since you are working with this very nice data about the variation of factor IX activity based on these specific residues, have you discussed, do you have any idea why it's not optimized for coagulation? Because maybe I know it's in the very important position in the coagulation cascade, so it looks like as if it's optimized for not being very effective. So that's the concern that I had.
Yeah, that is a great question. Something we have been thinking about. I think what's clear is that the factor IX activity that we consider normal, right, sort of the 50 to 150% normal, has to make this balance. So one, if you have too little of factor IX activity, you get hemophilia, and if you have too much, I think the data is pretty clear that you're at increased risk for thrombosis. And so I think factor IX has evolved to fit this happy medium. Sometimes we call this the Goldilocks effect, right? So it has to be not too hot and not too cold. It has to be just right. And I think that there's sort of different positions in the structure of factor IX that if you change one way, you can make it worse, meaning less activity. But if you change it, then there are other positions, if you change them, they can have increased activity. And it's had to do the balance of that. And I think the same thing is true for factor VIII. And it may be sort of true for a lot of proteins that can have pathology with too much or too little activity. Okay. And I also agree with you that your point that factor IX being high up on the coagulation cascade has to be more stringently regulated, because little differences in the activity get sort of magnified down the coagulation cascade.
Okay, thank you.
Gabi, você tem uma pergunta?
Oi, tem. Hi, Ben. So, do you, as for factor VII, you talked about those anti-drug antibodies. Do you think it has to do with the number of nucleotides that were changed?
Yeah.
And do you foresee any of this problem with this APC resistance factor VIII that you talked about?
Yeah. So I think it's intuitive that the more you change, the higher the risk of the immunogenicity is. And I don't think we know, and I don't think it's a phase transition where 1 is okay, but 3 is terrible. I think it depends on which amino acids there are. There are probably, we know this, that there are probably hot spots within different proteins that bind to MHC receptors differently, and that certain structural changes will then bind better and they'll be presented more. There's been a lot of sort of retrospective analysis of those factor VII variants, looking at how they bind MHC complexes both experimentally and then in silico modeling. And I think this is always an issue. And I think that one of the problems is that there's no sort of gold standard for assessing immunogenicity before you get to the clinic. And those variants went at phase 1, 2 without problems, and it's only when they expanded into the phase 3 that these anti-drug antibodies were noticed. But they were noticed in about 10% of the people that got them. I think our group's work with factor IX PADUA, I think, really establishes that its safety from an immunogenicity perspective, but there's also work doing the same type of in silico analysis. So I think if you can get away with one amino acid substitution, that's the best. I would just, I think what we know is that is PADUA now is the new baseline, so maybe one more might be okay. I think you want to be careful with every additional one you add. I think that will definitely be an issue with QQ, and I know that group is sort of thinking about doing some of the same experiments that were done for PADUA to sort of suss out that the potential immunogenicity of those positions. Interestingly, mouse, one of those arginines in mice, factor VIII is already glutamine, so it may be a little bit more flexible in that position. Though, they are in A2, which is historically a sort of a hot spot of factor VIII immunogenicity.
So, Ben, I have a question related to actually some ideas that Gabi just asked you, because, I think factor IX PADUA, there is no discussion that this is the best one for gene therapy for hemophilia B. And I agree your approach, seeing that maybe we can add and increase the specific activity. But the problem related to gene therapy is that the variability, we cannot predict the expression. And I think at the same time that we wanted to use a lower vector dose, but we can also predict that we will have an overexpression, as you know some patients that, and actually this is what happened with some protocols for the risk of thrombosis in the other hand. So how far you think we can go once we cannot predict the gene therapy expression at this point?
Yeah, so I think the variation in transgene levels post-gene therapy, affects sort of all the gene therapy programs for hemophilia. I think the rationale for trying to get the vector dose lower, for example, in the Spark hemophilia B trial that moved on to phase 3, sponsored by Pfizer, we saw 2 out of the 10 participants needed immunosuppression with steroids, and what the best type of immunosuppression is necessary, is an active clinical question. But I think if you can get to a dose where the rate of the capsid immune response or hepatotoxicity is such that you don't need to do steroids or very rarely, I think there's some benefit for that. I don't know if, and so like, what's that threshold? So, the Spark trial was carried out at 5e11 vector genomes per kilogram. In the earlier St. Jude's trial that had some expression, though low level, at 2e11, none of those participants developed capsid immune response. So I think, we sort of know, in a limited number of patients. So I think there is a sort of a goal line that could be investigated to have a little bit more potent vector to get below that threshold. I don't know how decreasing the vector dose will change the variability, but I think that's certainly something that has to be watched for, both as you decrease the vector dose, but then there's other people that want to increase the vector dose to achieve more, to put more participants into the curative range. So both need to be carefully watched, and I think it's hard to predict.
Yeah. And my other question is related to the related base foreign site. I think you showed something that's very interesting, because this is related more with the secretion of the factor VIII from the cell. And as you know, the elevations of ALT after gene therapy, of course, that can be part of the immunogenicity related to the factor, but it's not always can explain why some patients has this reaction. And one of the possibility is the unfold protein reaction. So I know that in the animal model, this is maybe quite difficult to evaluate, but do you predict that with this transgene, maybe we can also decrease this reaction and they have better outcomes with this most frequent side effects?
Yeah, so I think, so avoiding pace furin is one approach, the one that we have looked at or discovered to increase secretion. But there are a series of other variants of factor VIII that have also had the same thing. And in earlier work by Randy Kaufman and Steve Pipe with other variants, they pretty clearly showed that things that secrete better have less markers of endothelial stress. So I think that's a very attractive hypothesis. And anything that you can do to increase secretion, I think may address some of the hepatotoxicities, though I think it remains to be proven whether the hepatotoxicity, how much of it's related to non-protein Fulton response, but I think that's an attractive hypothesis as well. I think, the other interesting question is if these problems with secretion are because we're using hepatocytes, which are an ectopic site of factor VIII synthesis, and whether it would be different in other other tissue locations is also a really interesting thing about just factor VIII biology in general.
Yeah, totally agree. And what is your opinion about the evolutionary paper from Chris Doran? Can you comment? What is your impression about that?
Sorry, Margaret, what paper?
The importance of the data from the Chris Doran paper related to the evolutionary data. I showed the one figure from Chris Doran's at Emory University's recent Blood Advance paper
where they combined one of their ancestral factor IXs with PADUA and showed that it was about 3-fold higher factor IX activity levels after gene therapy in mice. And that's the third paper from that group using this technique in coagulation factors. They initially did it with factor VIII, and they have one with benwillibrand, and then the third one is factor IX. I think it sort of supports this underlying idea that Eric and I were talking, the first question about that factor IX, that human factor IX and human factor VIII have sort of evolved to have limited activity. And if you de-evolve, if you go back in evolutionary time, you often can find things that have a higher activity. I think the challenge is, that's a lot of, it's 90% similar to human factor IX, but that means there's 39 amino acids that are different. And so Gabriella asked me about, is it 3 or 6? We're talking 40 different ones for that variant. So I think how you translate that with so many amino acid substitutions is challenging.
Yeah, totally agree. Okay, anyone has another question for them? And thank you very much. I really appreciate your participation here. It was very helpful. I hope to see you soon, and I hope we have a chance for the future for you to be here in Brazil instead of this virtual model.
I hope to come to Brazil. Thank you so much for the invitation.
Okay, thank you very much.
Stay safe.
Okay, you too. Bye. Thank you. Precisa avisar o Michel, né?